Our lecture for today will be about cellular chemistry and the biological molecules. To understand the chemistry that allows biological events to give rise to life, we will begin by discussing the types of bonds that atoms can form one another. Molecular atoms are joined together by covalent bonds in which electron pairs are shared between pairs of atoms. The sample image on the screen shows how an atom looks like, where at the center is the nucleus with the proton and neutrons around it. The valence shells, also known as the electron shells or energy levels, are fixed distances from the nucleus of an atom where electrons may be found. The electrons in the outer shell are what you call your valence electrons. So to make an atom more stable, there should be around 8 electrons on the outer shell. The bond formation for covalent bonds are accompanied by energy release, which must be reabsorbed after breaking the bond, given that the energy required to break the bond is much greater than that of the thermal energy of the molecule. In many cases, two atoms can become joined by bonds in which more than one pair of electrons are shared. If two electron pairs are shared, as occurs in molecular oxygen or O2, the covalent bond is called a double bond. And if three pairs of electrons are shared, as in molecular nitrogen or N2, it is a triple bond. Quadruple bonds are not known to occur. The type of bond between atoms has important consequences in determining the shape of the molecules. For example, atoms joined by a single bond are able to rotate relative to one another, whereas the atoms of double bonds lack this ability. So, the more bonds there are, the more rigid a molecule will be. For the electronegativity and an equal or equal sharing of electrons, when atoms of the same element bond together, the electron pairs of the outer shell are equally shared between the two bonded atoms as in hydrogen atom. But if there are two unlike atoms that share the electrons, the positively charged nucleus of one atom, which is more electronegative, exerts a greater attractive force on the outer electrons than the other. Thus, the shared electrons tend to be located more closely to the more electronegative atom, and of atoms most often seen in biological molecules, nitrogen and oxygen are highly electronegative. This figure shows you a representation of the arrangement of electrons in a number of common atoms. Basically, the number of outer shell electrons is a primary determinant of the properties of an element. For polar and nonpolar molecules, in here we have water as an example of an element with covalent bonding. Water's single oxygen atom attracts more electrons forcefully than either of its hydrogen atoms. With this, it is considered to be a polar molecule. Such molecules have an asymmetric charge distribution, or you call a dipole. As a result, the OH bonds of water molecule are said to be polarized, such as that one of the atoms has a partial negative charge, which is actually the oxygen atom, and the other is a partially positive charge, which is endowed in your hydrogen atoms. Biologically important polar molecules have one or more electronegative atoms, usually oxygen, nitrogen, and sulfur. Molecules that lack electronegative atoms and strongly polarized bonds made up of carbon and hydrogen atoms are said to be nonpolar. Even the presence of strongly polarized bonds is of utmost importance in determining molecular reactivity. Next is ionization. Some atoms are strongly electronegative that they can capture electrons from other atoms during a chemical reaction. Just what, for the example that what we have right now is the formation of table salt with a combination of sodium and chlorine. So when you mix them together, they form table salt. But how does it happen? So a single electron in the outer shell of each sodium atom migrates to the electron deficient chlorine atom. As a result, these two atoms are transformed into charged ions. Because the chloride ion has an extra electron relative to the number of protons in its nucleus, it has a negative charge. and is termed an anion. The sodium atom which has lost an electron has an extra positive charge and is termed a cation. From there, you will be able to form your table salt. So just by looking at the example on the screen right now of how you can determine the difference of an ion and cation, it's more likely about the number of protons than your electrons. So you call it you call it a cation when it's positively charged and there are more protons than the electrons. It is now termed an ion if the atom is negatively charged and there are more electrons than the protons. Ions like sodium and chloride are relatively stable because they have filled an outer shell. A different electron arrangement in atom can produce a highly reactive species you call a free radical. So by checking on the image right here, you will see that a free radical does have an unpaired electron on its outer shell compared to a stable molecule. Non-covalent bonds govern interactions between molecules or different parts of a large biological molecule. Such bonds are typically weaker linkages, while covalent bonds are stronger. Non-covalent bonds depend on attractive forces between atoms having an opposite charge. Individual non-covalent bonds are weak, readily broken, but can be reformed easily. Moreover, non-covalent bonds can still mediate the dynamic interactions among molecules within the cell and can still provide considerable stable structures. For the first type of non-covalent bond, we have ionic bonds or salt bridges. This is a type of attraction between fully charged components. Ionic bonds are from transfer of electrons from one atom to another, leading to atoms with positive and negative charges that attract each other. Ionic bonds can hold molecules together, such as the DNA protein. In the DNA protein, it is negatively charged. Phosphate atoms are closely associated with the positively charged groups on the surface of a protein. Thus, the ionic bonds between them hold the complex together. Bonds between free ions are not that important in cells because cells are mostly water. Weak ionic bonds between oppositely charged groups of large molecules are much more important. Next, we have the hydrogen bond. Hydrogen bonds are known to be hydrophilic or water-loving. They also enhance the solubility in and interactions with water. When a hydrogen atom is covalently bonded to an electronegative atom, particularly an oxygen or a nitrogen atom, the single pair of shared electron is greatly displaced towards the nucleus of the electronegative atom, leaving the hydrogen atom with a partial positive charge. Hydrogen bonds in biological molecules that are in a solution can easily be broken because its energy, which is around 1 kilocalorie per mole, is similar in magnitude to its thermal energy. Next would be the hydrophobic interactions or water-fearing interactions. Nonpolar molecules such as steroids or fat molecules are essentially insoluble in water because they lack the charged regions that would attract them to the poles of water molecules. When nonpolar compounds are mixed with water, the nonpolar or hydrophobic molecules are forced into aggregates, which minimizes their exposure to the polar surroundings, calling it as a hydrophobic interaction. Most believe that they are not true bonds since not usually thought of its attraction between hydrophobic molecules, but an energetic drive. A classic example is that when you are cooking meat like chicken or pork, the droplets of the fat molecules rapidly reappear on the surface of the beef or chicken soup even after the liquid is stirred with a spoon. So that's how the hydrophobic interaction happens. Next, we have the van der Waals interactions, which uses forces. So hydrophobic groups can form weak bonds with one another based on electrostatic interactions. This is due to the slight perturbations of electron distributions. So meaning there is an asymmetry or imbalance when it comes to the electron distribution. So if two of such molecules are very close together and appropriately oriented, two electrically neutral molecules will experience weak attractive force bonding them together, which is what we call the van der Waals forces. So basically, unlike the connection of your table salt, which is sodium and chloride, which has a positive and a negatively charged atom, with the van der Waals interactions, most likely the charge of both atoms are in neutral. Okay, so none of them are positive and negative. Both are usually neutral in charge. The life-supporting properties of water. Life on Earth totally is dependent on water, may it be plants, animals, humans, and maybe life anywhere in the universe as well. So the unique water structure are or does have the following properties. So first, it is highly asymmetric. So it has oxygen at one end and two hydrogens at the other end. Next, it has two highly polarized covalent bonds, and it is very adept in forming hydrogen bonds. So when you say adept, it is more likely to be very active to form hydrogen bonds. Next, the tendency of water molecules to adhere to each other is evident in water's thermal properties, and it is also a good solvent. So it dissolves many things like your solutes more than any other solvent, but it is inert itself. Next, let's discuss now about acid bases and buffers. Basically, acids and bases exist in pairs or in couples. So later on, I'll be discussing further how are they considered as couples. So first, let's define acid. So an acid is a molecule able to release or donate a hydrogen ion to a medium through the process of dissociation. So when a proton dissociates and is released into a medium whenever a hydrogen atom loses an electron. And then the base is a molecule capable of accepting a hydrogen ion, usually your protons. So you also have what you call your amphoteric molecule, which is a molecule that can serve as both an acid and a base, usually both a positive and negative charge. And basically, water and amino acids are actually examples of your amphoteric molecules. So how do we say that acids and bases are actually couples? So basically, acids actually vary greatly in the ease with which they give up protons. So when the acid loses a proton, it becomes a base, which is termed conjugate base of the acid. Similarly, when a base accepts a proton, it forms an acid, which is termed as a conjugate acid of the base. So if a proton is readily lost, attraction of a conjugate base is slower and acid is stronger. For example, your hydrochloric acid. And then it readily transfers its proton to the water. A strong acid's conjugate base, for example, your chloride, is a weak base. And in here, the hydrogen dissociates since water is a stronger base. And next, we have your weak acid like your acetic acid, which is most likely undissociated in water because acetate ion is stronger base than your water. Next is pH. So pH is the measure of hydrogen concentration computed as negative log of hydrogen, where hydrogen is the molar concentration of protons. In logarithmic scale, this is actually the increase of 1 pH unit, meaning 10 times increase in hydroxide or 10 times decrease in hydrogen. So the formula for dissociation of water into a hydroxyl ion and a proton would be 1 unit of water yields to hydrogen plus hydroxide. Or more accurately, if there are 2 units of water, the yield would be a hydronium plus hydroxide. So in pure water, the concentration of both hydrogen and hydroxide is approximately around 10 to the negative 7 moles. Pure water thus has a pH of 7, and since we view water as being the standard solvent into which other molecules are dissolved, it is common to refer to pH 7 as neutral. So low dissociation indicates water is very weak acidic. Next, most biological processes is sensitive to pH change since pH affects biological molecule ionic states. So in here, we have an example of the amino acid R group or side chain that acquire charges. So we have here a carboxylic group or amino group from amino acid. As the hydrogen ion concentration increases, it becomes protonated, which can disrupt the activity of the entire protein. So when you say proteinated, you are acquiring additional charge. So from this side chain, okay, it becomes negative, and this one becomes positive. So even slight pH changes altering these groups can disrupt shape and activity of the entire protein and can impede biological reactions. Next, we have the buffer. So the buffer minimizes pH fluctuations and resists changes in pH. So they bind or release and reacts with hydrogen and hydroxide ions depending on conditions. So thus, they protect organisms and their cell. Usually, they contain weak acid with its conjugate base. An example that we have right here is the blood. So take a look at this simple formula containing the bicarbonate ion and the carbonic acid. So during exercise, an increase in the hydrogen ion concentration will have the bicarbonate ions to bind with the excess proton, removing them from the solution. And then, when there's an excess in hydroxide ions during hyperventilation, they are neutralized by protons derived from your carbonic acid. Hence, buffering actions by the carbonic acid and bicarbonate ions normally hold blood pH at about 7.4. So you see how buffer works to maintain homeostasis. So these two ions are very, very important when it comes to regulating the pH of the blood. And then pH of fluid within cells are actually regulated by the phosphate buffer system that includes your dihydrogen phosphate and hydrogen phosphate. Next, let's tackle now the nature of biological molecules. First in our list would be the organic molecules, which were found in cell dry weight consisting of carbon atoms. So chemists thought once that they can only be found in living organisms, and their name distinguishes them from the inorganic molecules found in the inanimate world. However, as chemists learn to synthesize more and more of these carbon-containing molecules in the lab, the mystique associated with organic compounds disappeared. The compounds produced by living organisms are now what you call your biochemicals. So next, we have organic chemistry. So with organic chemistry, it's very important that you know the use of carbon. Okay, so carbon, both its size and electronic structure, allow carbon to generate many molecules. So there are around 100,000 known usage of this carbon. So basically, the main thing about carbon is that it can bind up to 4 other atoms since it has only 4 outer shell electrons. And basically, you needed to fill 8 electrons in each shell. And then it can form carbon-containing backbones with long chains, which may be linear, branch, or cyclic in form. Okay? And carbons can also be connected by single, double, or triple bonds. So with double bonds, you have, or you can link it with oxygen and nitrogen, and with triple bonds with nitrogen. Okay? So compounds are very stable since strength of covalent bond inversely proportional to atomic weight of elements are involved. Example, you have the silicon, which is just below the carbon in your periodic table. Next, we have the hydrocarbons. So they contain only hydrogen and carbon atoms, and they are considered to be the simplest group of organic molecules. As more carbons are added, skeletons increase in length and structure becomes more complex. So the most common type of your hydrocarbon is the ethane, which has the formula of C2H6. So it is also fully reduced or saturated when each carbon bound to maximum number of hydrogen atoms. And unsaturated compounds have double or triple bonds, and they lack maximum number of hydrogen atoms. There's also rotation of carbons around single bonds, but not around double or triple bonds. Next would be the functional groups. They are particular atom groupings that often behave as a unit. They are responsible for physical properties, chemical reactivity, and solubility in aqueous solutions. And basically, they replace hydrogens in your hydrocarbons. So, hydrocarbons do not occur often in living cells, although they form the bulk of fossil fuels formed from the remains of ancient plants and animals. And here, some of the major functional groups are listed. You have the hydroxyl group, you have the carboxyl group, and it can actually acquire charge. So carboxylic acids reacts with alcohols to form ester bands. And then sulfhydryl group, or the SH group, reacts to form disulfide bonds with polypeptides. And then the amino group, which is the amide functional group, it acquires charge as well and reacts with carboxylic acid to form your amide bonds. Here you will see the list and a table of how the functional groups look like. So you have the methyl group, hydroxyl, carboxyl, amino group, phosphate group, carbonyl, and sulfhydryl. And then below here, you will see how the ester bonds and amide bonds look like. Okay? So be familiar with these functional groups. Next would be the functional classification of biological molecules. The first one would be the macromolecules. So macromolecules forms the structure and carry out activities of the cells, usually huge and highly organized molecules, and they contain from dozens to millions of carbon atoms. Because of their size and the intricate shapes they can assume, some can perform complex tasks with great precision and efficiency. Endow organisms with properties of life and set them apart chemically from inanimate world. This is actually divided into four major categories. First, you have the proteins, the nucleic acids, polysaccharides, and the lipids. So the first three, proteins, nucleic acids, and polysaccharides, are actually polymers. They are made of large number of low molecular weight building blocks you call your monomers. And basic structure and function of each type of macromolecule are similar in all organisms. So the organic molecules commonly found within living cells can be divided into several categories based on their role in metabolism, and these macromolecules are constructed from the monomers by a process of polymerization. Next would be macromolecule as building blocks. So most macromolecules in the cell have short lifetime compared with cell, except the DNA. So these are steadily broken down and replaced by new macromolecules. Most cells contain supply or pool of low molecular weight precursors to build macromolecules. And we have the monomers, which are the building blocks of macromolecules. So they can be in the forms of sugar or polysaccharides. They are also in the form of amino acid for proteins. You have the nucleotides for nucleic acids and fatty acids, and glycerol for your lipids. Next, metabolic intermediates, or what you call the metabolites. These are molecules in cells that have complex chemical structures and must be synthesized in step-by-step sequence, beginning with the specific starting materials. In a cell, each series of chemical reaction is called a metabolic pathway. And compounds formed along pathways leading to the end products might have no function per se except as a stop on the way to the end product and are called metabolic intermediates. Next would be the molecules of miscellaneous function. So these are vast bulk of cell dry weight, which is made up of macromolecules and their direct precursors. So the molecules of miscellaneous function include such substances as vitamins that function primarily as adjuncts to proteins. You also have certain steroids or amino acid hormones. There are also molecules involved in energy storage like your ATP or adenosine triphosphate and creatine phosphate. They are also involved in regulatory processes such as your cyclic AMP and for metabolic waste products such as your urea. Next biological molecule is the carbohydrates. So carbohydrates, or what is also known as glycans, comprise of a group of substances including simple sugars or monosaccharides and larger molecules made from them. So they serve primarily as chemical energy storehouse and durable building material for biological construction. And most of them have the general formula of CH2O. So sugars containing 3 carbons are known as trioses. Those with 4 carbons are tetroses. Those with 5 carbons as pentoses. Those with 6 carbons are hexoses. And those with 7 carbons are heptoses. So the N values for that range is from 3 to 7. The structure of simple sugars. So each sugar molecule consists of carbon atoms that has a backbone linked together in a linear array by single bonds. So each carbon of backbone is linked to single hydroxide group except for one bearing carbonyl group. Sugars with 5 or more carbons convert by self-reaction into closed ring-containing molecule with hydrogens and hydroxides above or below the ring, and the ring is not planar but in the 3D conformational form resembling a chair. So this image shows you the different structures of your sugars. So if a carbonyl group is located in the internal position, this is actually to form a ketone group. So the sugar is now known as a ketose, and such as an example would be your fructose. Okay? But if the carbonyl group is located at one end of the sugar, it forms an aldehyde group, and the molecule is now known as an aldose. So all of the carbonyl that are located on the edge or at the end of a conformation, they are considered to be aldoses. So examples B, C, D, E, and F are all aldoses. Next, we have stereoisomerism. This is the arrangement of groups around a carbon atom, which is depicted with carbon in center of a tetrahedron with bonded groups of projecting into its 4 corners. So with 4 different groups attached to the carbon, it can exist in 2 configurations that cannot be superimposed on one another. So the 2 configurations are mirror images. Usually you call them stereoisomers or enantiomers of each other. So if the hydroxyl group of a carbon projects to the right, the aldose is a D sugar. But if it projects to the left, it is what you call an L sugar. So straight chain glucose converts by self-reaction into six-membered pyranose ring with carbon 1 being asymmetric. Next, linking sugars together to make a larger molecule. So bond joining sugars together is what you call a glycosidic linkage or bond. So its conformation is COC. The forms by reaction between C1 of one sugar and hydroxide of another. Sugars can be joined by a variety of different glycosidic linkages. So two monosaccharides covalently bond together to form a disaccharide. So they serve primarily as readily available energy stores. You also have oligosaccharides, which are small chains of sugar, usually attached to lipids and proteins, converting them to glycolipids and glycoproteins respectively. And then you also have what you call your polysaccharides, which are composed of many sugars hooked together and they form very large molecules. The different types of polysaccharides includes your sugars, starches, cellulose, chitin, peptidoglycan, and glycosaminoglycans. So glycogen is a branch glucose polymer mostly joined by alpha 1 to 4 glycosidic bonds. And then you have the starch, which is a glucose polymer. It is a mixture of two different polymers, including the amylose and amylopectin. And plants bank their surplus chemical energy in the form of starch, like in potatoes and cereals are actually made up of starch. Okay? And then you have cellulose, which are tough, durable structural materials like what you see in cotton and linen, and they are the major plant cellulose components. Next, you have the chitin, which are unbranched polymer of N-acetylglycosamine, the amino group instead of hydroxide on glucose. So they occur widely as structural materials among invertebrates, particularly in the outer covering of insects, spiders, and crustaceans. So the chitin is tough, resilient, yet flexible material, not unlike plastics. So insects owe much of their success to this highly adaptive polysaccharide. And then lastly, we have the glycosaminoglycans or the GAGs. They are repeating disaccharides. So you have their two different sugars alternating and they occur in spaces surrounding cells. Next, we have the lipids. So lipids are a diverse group of nonpolar biological molecules whose common properties are their ability to dissolve in organic solvents, such as chloroform or benzene, and their inability to dissolve in water. Lipids of importance in cellular function include fats, steroids, and phospholipids. So basically, these lipids are principally composed of carbon, hydrogen, and oxygen. They are not considered macromolecules, but they are aggregates to form large complexes. Triglyceride, also known as fats or triacylglycerol, consists of glycerol molecule linked by ester bands to three fatty acids. So they are used to serve as lipid storage for fuel or stored in adipocytes. Next, we have fatty acids. They differ from one another in the length of their hydrocarbon chain and the presence or absence of double bonds. Fatty acids present in cells typically vary in length from 14 to 20 carbons. Fatty acids that lack double bonds, such as stearic acid, are described as saturated. However, those possessing double bonds are called as unsaturated fatty acids. Next, we have the sterols and steroids. They are complex and they are characterized by four-ringed hydrocarbon structures. So there are 4 joined rings that differ in number and position of double bonds and functional groups. So examples would be cholesterol, which is the most common and one of the most important type of lipid. It is a component of animal cell membrane, but not in internal membranes or in plants. Next, we have the adrenocortical hormones. Such examples are your aldosterone and cortisol. So basically, these are products of your adrenals. And then next, you have the sex hormones. They are actually precursor for the synthesis of some steroid hormones like your estrogen, progesterone, and your testosterone. And then next, we have here vitamin D3 and the bile acids, which are involved in the lipid digestion in the intestine. Next, we have the phospholipids or the phosphoglyceride or diacylglycerol. So it is composed of glycerol plus 2 fatty acids plus a phosphate group on the third hydroxyl. So it is often an amino group as well. It is highly charged at the physiological pH, and it is also considered as amphipathic. So the major cellular function of this one is of course, we all know this, the presence in membranes. So properties of which depend on the phospholipids. Usually, it has a hydrophilic alcohol attached to phosphate like choline, ethanolamine, inositol. Only one lacking charge amino group, and with just phosphate group called a phosphatidic acid. It also usually have 16 and 18 carbon fatty acids, one saturated and one unsaturated fatty acid chain, and the length and degree of unsaturation affect membrane fluidity and are regulated. So next we have the proteins. So it is composed of hydrogen, carbon, oxygen, and nitrogen, and usually sulfate or phosphate. So they are very large macromolecules and they're polymers of amino acids. So their traits and functions are more varied than other organismal molecules. So they can be for enzymes, they can be used for structural uses or both. So they execute almost all cell activities. Typical cell may have around 10,000 different ones with high specificity. So properties of the R groups or its side chain are determined as inter or intramolecular interactions that determine molecular structure and protein activities respectively. So give amino acid, it actually gives the amino acids their variability. Next would be the 4 amino acid and R group categories. So these are classifications of the side chains or R group characteristics. And not all of the amino acids are found in all proteins, nor are various amino acids are distributed in an equivalent manner. Okay, so the first category would be the polar charge. So this contains a side chain that becomes fully charged. That is, the side chain contains relatively strong organic acids and bases. And this includes aspartic acid, glutamic acid, lysine, and arginine. The second type would be the polar uncharged. The polar uncharged do have side chains of these amino acids have a partial negative or positive charge and thus can form hydrogen bonds with other molecules including water. These amino acids are often quite reactive. Included in this category are asparagine and glutamine, threonine, serine, and tyrosine. Next, we have the nonpolar category. In here, side chains of these amino acids are hydrophobic and are unable to form electrostatic bonds or interact with water. So it includes alanine, valine, leucine, isoleucine, tryptophan, phenylalanine, and methionine. And lastly, the other 3 amino acids, there's actually a difference in their conformation. So for glycine, it has a side chain of only a hydrogen atom and glycine. For proline, it is unique in having its alpha amino group as part of the ring. And then cysteine contains a reactive sulfhydryl group and is often covalently linked to another cysteine residue as a disulfide bridge. So those are the differences of these amino acid categories. So here are the conformation or the arrangements of the different amino acids. So how the properties of the side chains look like. So you have there the polar charge, the polar uncharged, the non-polar, and of course the side chains with the unique properties. Okay. Next, information about proteins. So not all of your amino acids are found in all proteins, nor are the various amino acids distributed in an equivalent manner. And then a number of other amino acids also seen in chains like thyroxine, hydroxyproline, or hydroxylysine. Also, the character of amino acid R groups or side chains can be ionic, polar, or nonpolar is very important to protein structure and function. Thus, the side chains also affect solubility. So, amino acids can be separated on the basis of your solubility process. Next, we have the conjugated proteins, which are involved or they involve another type of molecule attached covalently or non-covalently to protein, and they include the following. First, you have the nucleoproteins. These are a combination of protein and nucleic acids. You also have the lipoproteins, which are made up of proteins and lipids. You also have the glycoproteins for proteins and carbohydrates, and the last one would be various low molecular weight materials like metals and metal containing groups often attached. And then proteins are a good illustration of intimate relationship between form and function. So the structure of most proteins is completely defined and predictable. Each amino acid in one of these giant macromolecules is located at a specific site with the structure, giving the protein the precise shape and reactivity required for the job at hand. And then protein structure described at several levels of organization. So each emphasizes a different aspect and each is dependent on different types of interactions. So for the four levels of protein structures, we have the primary, secondary, tertiary, and quaternary. So for the primary protein structures, the sequence of a chain of, it's just a sequence of chain of amino acids. Okay. So for the secondary protein structure, hydrogen bonding of the peptide backbone causes the amino acids to fold into a repeating pattern. So in here, you will see that there's already the formation of pleated sheets and the formation of the alpha helix. Okay. And then for tertiary proteins, this is already a three-dimensional folding pattern of a protein due to the side chain interactions. And then lastly, we have the quaternary structures that happen when subunits of proteins or multiple polypeptide chains come together. So that's how the different proteins can be structured. So I would like to give an example of protein. So for example, the hair. Okay, so for you to be able to easily understand how proteins are being folded. So for example, if you were born or if you do have a straight hair, more likely you have a primary type of protein structure. Okay, but if you're the type of for example, you have a wavy hair, there's a possibility that you may have a secondary type of protein structure in your hair. Okay, but if you are the type that your hair is very curly, okay, so it can be um an example of a tertiary or quaternary proteins depending on how curly your hair is. Okay, so that's that's a simpler example of how you can think of the different protein structures. Okay, so next we have the nucleic acids. So these are primarily involved in the storage and transmission of genetic information and may also be structural or catalytic. So there are two types in living organisms. We all know this one, which is the deoxyribonucleic acid or the DNA, and the ribonucleic acid or RNA. So basically, DNA serves as genetic material of all cellular organisms, and RNA plays that role in many viruses. And information stored in DNA is used to govern cell activities through the formation of RNA messages. So nucleic acids are constructed as long-chain strands of monomers called nucleotides. And concentrate here on RNA as representative molecule. They are more complex, double-stranded DNA. So both DNA and RNA are composed of nucleotides. You have your phosphate, you have the sugar, and then you have the nitrogenous bases. So they are connected to form polymers you call your polynucleotides. So the phosphate group is technically characterized with the presence of phosphate linked to the 5' carbon of the sugar. And then the 5-carbon sugar can be a ribose or a deoxyribose. And then the nitrogenous bases can be a pyrimidine, which is a one-ring base, or a purine that has two rings on it. Okay? So the sugar and nitrogenous base together form a nucleoside. So nucleotides of RNA strand are known as ribonucleoside monophosphates. The phosphate is normally linked to the 5' carbon of the sugar, and the nitrogenous base is attached to the sugar's 1' carbon. So monomers polymerize when sugar from the 3' hydroxide is linked by ester bond to 5' phosphate of the next nucleotide in the chain. So 3' to 5' phosphodiester linkage is present. So nucleotides are joined by sugar phosphate linkages. In this image that you see here, you will see the different nitrogenous bases in nucleic acid. So for purines, you have guanine and adenine, and then for pyrimidines, you have the cytosine and thymine. But if this thymine undergoes translation process, or transcription process, it will become now your uracil. So general structure and function of DNA or RNA. So sequences of bases determine specificity of DNA or RNA and encodes hereditary information for synthesis of proteins. RNA is usually single-stranded but can fold back on itself to form frequent double-stranded regions with local hydrogen bond pairing, just like in DNA, and complex three degrees structure. So types of RNA includes ribosomal RNA, transfer RNA, and messenger RNA. So rRNAs do not carry genetic information but serve as structural scaffolds to which ribosome proteins can attach. They recognize and bind various soluble components used in protein synthesis. Some RNAs have catalytic activity, for example, the ribozymes or RNA enzymes. They are double-stranded regions held together by hydrogen bonds between the bases and also hold together DNA strands. ATP or adenosine triphosphate is an RNA nucleotide. It is a source of energy for biochemical reactions, and it is most used at any given moment in any living organism when it is derived from it. So that actually ends the second lecture for our class. So the reference material would be still Cell and Molecular Biology and Concepts and Experiments by Karp et al. And the images, some of the images were actually lifted from google.com. So thank you very much for listening, and I'll see you on our next lecture. Thank you.